Je comprends pas pourquoi il veut pas me partager seulement l'écran.
Ok, c'est bon, c'est parti pour le stream. Les gens ont le lien. Comment on lui file le lien?
C'est bon, de toute manière, mets-le en public, comme ça ils iront directement sur la page NPM. Pas besoin de ce lien-là. Confidentialité.
Ouais, je gère de ton côté.
Oui, si jamais. Je comprends pas exactement.
Ouais, mais c'est normal, c'est pas grave.
T'as moyen de te connecter sur Chrome, sur celui-ci, parce que j'arrive pas à...
Là, en dessous, partager.
Ouais, mais il me met connexion encore.
Ah mince.
J'ai essayé. Quoi?
Ça marche? Non, il y a...
On essaye.
Pour YouTube,
mais je vais essayer sur Chrome. En fait, j'avais fait avec celui-là. Alors, sinon, au pire des cas, on fera tout avec celui-là, mais le problème, c'est que j'avais un peu peur que celui-là, il crache si jamais on diffuse sur YouTube en même temps et diffuser ça. Mais je vais commencer par regarder si ça fonctionne sur Chrome.
Ouais, sur mon Discord, ouais. Je vais là aussi. Ouais. Je te l'envoie. Très bien.
C'est cool, hein.
Tu veux mon Discord? Non, mais je pense que c'est plus.
Information de la salle.
C'est bon, tu t'en sors.
Sur les informations de la salle, il faut que j'essaie de me connecter via Chrome.
Comment?
Est-ce que je peux être les... Est-ce que tu peux faire un meeting?
Ouais, mais c'est bon. Ça ligne. Je pense que c'est possible. C'est un peu comme ça.
Je fais formation.
Ah, tiens, il n'y a pas un fois. Ah non, il y a un point.
Oui, j'ai vu.
On arrive.
Le nom, il va finir. Ah ouais, c'est pas ça.
Oui.
Enfin, forcément. Comment ça veut dire?
T'as des...
Ah oui, non.
Ah ouais, attends, j'allais reprendre le lien.
Ouais. Là, dans information. Information. Longueur envoyée.
Vas-y, mais... Tu vas passer?
Stop vidéo, non?
Parce que je vais share du coup à partir de là. Il faut que j'arrête de share ici. Attends, je vais me déconnecter.
Si ça marche quand même. Je vais me connecter à la gros voilà,
Yes.
il m'a envoyé le lien. Donc...
Ah.
Pas mal. Jean-Marc, tu nous entends? Ouais, c'est bon. Par contre,
Donc on va bientôt démarrer là. Ouais, très bien. On est tous là, mais dernier réglage technique et puis ça démarre.
Ok, super.
Je regarde.
Ok. So, well, it's fine. Sorry for
Tu peux m'envoyer le lien,
inconvenience.
le lien Webex juste.
Jean-Marc, you can hear us?
Jean-Marc, could you hear us?
Yes, yes.
Okay for you?
Yes, perfect.
Okay. So, welcome to everybody. We are here for the PhD defense of Xavier Roland. So I'm for today the president of this jury. I'm Michel Doja, research director at INSERM, and I work at the Grenoble Institute of Neuroscience. And for this jury, we have from the left, Madame Sorina Camarasu-Pop, research engineer at Creatis at Lyon. Then you, Jean-Marc Jezekel, professor des university professor here at INRIA, at University of Rennes. Camille Momet, supervisor of this PhD thesis and research scientist at INRIA. And Pierre Morel, also the supervisor of this PhD thesis, and who work here at University of Rennes, and then, right, and Daniel Margulies, research director at CNRS, at Integrative Neuroscience and Cognition Center at Paris. So, Xavier, you have 45 minutes for approximately to defend your work.
Okay.
So, you could start.
Thank you. So, I thank everyone for coming here to assist to my PhD defense. So, I will present the works that I have done under the supervision of Camille Momet, Pierre Morel, and Christian Barillot at the beginning of my thesis about the impact of analytical variability on data compatibility in fMRI studies. So, first, I will present a bit of context by introducing the issue of the reproducibility crisis in neuroimaging, how we consider the solution of taking advantage of data sharing to reuse data and the situation of data combination that we want to address, and how analytical variability in fMRI pipeline can be an issue in this situation. So once we have presented the context, we will present our contributions, which are about the impact of analytical variability on data compatibility when performing group analysis using data processed differently. Our first contribution will be about the estimation of this pipeline effect, and the second one about their correction. So first, for a bit of context, first I will introduce neuroimaging. So neuroimaging is a field of neuroscience which takes advantage of neuroimaging techniques to make in vivo observation of the brain. There are a variety of brain imaging techniques, such as electroencephalography or magnetic resonance imaging, and they can be used for various types of cases. For example, for MRI, magnetic resonance imaging, there is structural imaging, which is used to make observation about anatomical features of the brain, and functional imaging, where we see variations over time of signals and see how these variations of signals are related to brain activity and to brain functions. As we say, there is a variety of techniques and a variety of use cases, and for the case of functional MRI, we focused on what is called test-based bold fMRI. So a short explanation. So in test-based bold fMRI studies, subjects participate in an MRI session, and while they are in our MRI, temporal sequence of images of the brain are acquired. These images of the brain consist of signals with various intensities at each position of the brain, and these signals correspond to blood oxygen level-dependent signal, because we take advantage of the fact that brain activity is related to increase in cerebral blood flow. So once we have these images, we have at each position of the brain, for example in this case, an MRI signal over time for each subject. And the thing is that while the subjects are in the MRI machine, they are asked to execute tasks depending on a paradigm, and this paradigm depends on what sort of function we want to study. There are motor paradigms, there are language paradigms. And then, once we have the data, we observe how the signal is related to the various tasks performed at each time in the MRI machine for each subject. Now that we have introduced a bit the context of the type of work that the domain in which we want to work, we will introduce the issue of the reproducibility crisis. So, in experimental science, reliability of experiments is very important because it is used to, these experiments and the research results that are found upon them are used to make, to create further research and to find further research results. So these results need to be reliable, and to be reliable, we need to ensure that reproducing the experiments that we did to obtain the results will yield the same results. And this is one of the main problems in experimental science today in a variety of experimental science fields. We found that a number of published scientific research findings cannot be reproduced. This problem was essentially highlighted over the last 10 years, but before that, it was shown that there were various factors which could undermine the reliability of scientific findings in the way research is done and undermine their reproducibility. One example of study which highlighted this problem of reproducibility was the reproducibility project in psychology, where 270 teams were asked to try and reproduce results from 100 published scientific studies in the field, and 39% success rate only was found, which means that all the other analyses failed to be reproduced. Since this reproducibility crisis is an important problem, various causes for this crisis have been highlighted. The first one is the question of the lack of data and protocol description in experimental science studies. The first thing that we want to do to ensure that the results are reliable is that we may use the exact same data and exact same protocol to perform the exact same experiment and try to find the exact same results, so that we can ensure that there is no fraud nor errors in the way the research was conducted. However, it was found that a large number of studies did not share the material which is necessary to allow to reproduce the exact same experiment, either lack of the data or the lack of description of the experimental protocol. Another one was the question of the publication bias. So as we say, in experimental science, there are various types of data and protocol that we can use to perform for some research question, and this variety of possibilities leads to a variety of possible outcomes. More specifically, we can find negative results and positive results for a same research question. And one problem in the way science is done is that published results is that in journals, there is a tendency to publish essentially positive results. This means that for the same research question, if there is a variety of studies which find both negative and positive results, mainly the positive results will be, another problem which is related to that is that of analytical flexibility. As we said, there is a variety of possible protocols, and this variety of possible protocols, the higher we have a number of possible protocols for the same study, the higher we have, the higher the number of possible outcomes is, and the higher the probability of finding at least one positive result, one detected effect when there is none, the higher this probability is. And finally, the main problem that we want to address is the question of low statistical power. When we want to detect an effect, there are various factors which enhance the probability of detecting this effect. The first one is the effect size. The higher the effect size, the higher the probability. But there is also the problem of sample sizes. When sample sizes are too low, there is a lower probability of detecting existing effects. So that will mean that we will have a lower share of true positive results which will be detected. And so among results detected as positives, the proportion of false positives will be higher. And this is what we want to address in our work. So in the field of neuroimaging, more specifically. Here we see the evolution of sample sizes and the associated detectable effect sizes at 80% statistical power. So of course, the higher the sample sizes are, the lower the necessary effect size to detect the effect is. We see that in the last decades, we had numbers which were considerably low in terms of sample sizes. And although we can see that there is an increase and also an increasing number, because here this is a logarithmic scale, an increasing number of very large scale projects with a very large amount of data, we can see that over the last years, we still had insufficient sample sizes. And this is the question that we want to address. How could we simply have higher sample sizes in neuroimaging studies? So what we wanted to do was take advantage of the increasing trend toward data sharing in neuroimaging. So data sharing has recently developed in neuroimaging, mostly over the last decade, but it started in the early years 2000 with the first major attempt for data sharing being the fMRIDC, although it was not very well received at the time because data sharing was not encouraged because of various reasons such as privacy and ethics reasons. The possibilities that it showed led to an increasing number of projects for data sharing. This includes repositories where researchers may share their data, may make them available to the public, such as INDI or Open Neuro. There are also projects which are funded specifically for data sharing. So there are studies or projects for which a very large amount of data is acquired for a very large amount of subjects and participants. And this data is shared so that researchers can reuse this very large amount of data for further studies. This is through the ANLI or the Human Connectome Project or UK Biodonc. And finally, there is a question of densely sampled data sets, where these are projects where there will be a certain amount of subject data available, but for each subject, a very large array of data will have been sampled. So because of this very large amount of available data today, we think that it may be interesting to take advantage of data sharing to reuse the data. And one possible case is a case where we have relevant data in various data sets. So we may have, for example, for a study, some data for healthy subjects or in subjects in separate data sets, and we may want to combine them. However, there is an issue which appears here, which is that in neuroimaging, before performing group analysis which combine the subject data, the subject data are processed using subject-level processing pipelines. In standard fMRI study, as is done here, all subjects are processed using a same subject-level processing. All subject data are processed using a same subject-level processing pipeline. However, if we combine data from various data sets, these various data sets may use different pipelines to process the subject data. One data set has a specific pipeline, another data set has another specific pipeline, and we may find ourselves in a situation, where we will have various subjects which are processed differently, with data are processed differently, and for which we will perform group level analysis with we defined these processing differences and the variability in outcome which may result from these processing differences as analytical variability, which is the variability resulting from the different possibilities in terms of experimental methodological choices for the processing and analysis protocol. There is a variety of aspects of analytical variability which have already been studied in the literature. One example of recent study which observed the issue of analytical variability was NAPS, where one data set was given to 70 different teams of experts who performed analysis to test 9 ex-ante hypotheses, and for 5 of these hypotheses, results were inconsistent across teams. This is not the only study which was done on the subject. Various studies have studied specific aspects, as we said, and specific components to answer specific questions. One aspect which was studied was the question of the differences in terms of pipeline choices for the processing steps applied in the pipeline, processing analysis steps, because in pipelines there is a variety of processing steps for which there is a variety of methodological options, whether we perform or not certain operations, in which order we perform them, and what parameters we use for each parameters or algorithm we use. Another one was a question of software packages. As we said, there is a variety of processing choices. There is also a variety of software packages available, such as FSL, AFNI, or SPM, which cover about 80% of the literature in fMRI. And across these software packages, the default choices, the available choices for algorithms and parameters may vary, which leads to variety in results if we use different software packages. And finally, there is also the question of operating system conditions, which may lead to numerical errors in different operating system versions, and which can give substantial differences in the outcomes, in the results. We saw that there were various studies which observed the question of analytical variability, but they essentially focused on a situation where we study variability across analysis. As we said, in standard fMRI analysis, all subject data are processed with the same pipeline, same subject-level pipeline, before group analysis. And this was what was done in most of these studies, which is that they perform one analysis where all the subjects are processed the same way before group analysis, but they perform this different times, each time with a different pipelines. As we say, the situation that we want to address is one situation where we combine data processed differently, and this is how we want to address the issue of analytical variability by studying it where it appears within analysis. When within a same group analysis, various subjects are processed using different subject-level pipelines. And so before going to my contributions, I will quickly give an overview of fMRI pipelines and how analytical variability may appear in the pipelines. As we say, there is subject-level processing on subject data before performing group-level analysis, which in fMRI is called second-level analysis. This subject-level processing is composed of pre-processing and subject-level statistical analysis called first-level analysis. Pre-processing consists in applying correction steps, correction operations on the data to make it usable for first-level analysis. To adapt it for first-level analysis, this includes steps such as motion correction, registrations toward a template space, and also smoothing, which is one parameter which will interest us, for which we can use various parameters such as we can use a parameter value which is the smoothing kernel full width at half maximum, which can take different values. So here, for example, we have the same image smooth with two different smoothing kernels with different full width at half maximum. And also, as we said, once the data is pre-processed, we can perform subject-level statistical analysis. As we said in the beginning, what we want to do in task-based fMRI is to see the relationship between ball signal in time at each position of the brain and the tasks which are performed by the subject in the MRI machine. So to do so, we use multiple linear regression using a design matrix whose regressors are task regressors. These task regressors are the stimuli time series associated to each task, convolved with a function called the hemodynamic response function, the HRF, which is used to model the expected response in case of stimuli in fMRI. And we fit the ball signal to this design matrix, and finally, parameters are obtained for each task regressors, and this can be combined into linear combination, which is called a contrast, resulting in a specific contrast map as the outcome of first-level analysis. And we have other possible options for first-level analysis, which are the use of temporal derivatives of the HRF to correct to do time adjustments for the response to the stimuli, and adding motion regressors. So as we said, we can perform motion correction at preprocessing. At motion correction, 6 motion regressors are estimated, and they can be included also in the design matrix to account for the correction of the effect of motion correction at first-level analysis. We can also use their derivatives, their squares and the squares of derivatives, to go up to 24 motion regressors added in the design matrix. So these are the parameters from which there are some of the parameters from which we can have variations, and for which we observe the effect of variations in the case that we introduced for our study of analytical variability. So this is what we are going to present now, our first contribution, which is about the impact of analytical variability on data compatibility when combining data processed differently in between group analysis in fMRI. So we say that, to remind of what we said previously, we say that we want to combine data to increase sample sizes to have a higher chance of detecting true positive results. However, we have one problem, which is that in fMRI we can also have false positive results, and our question is whether or not combining data processed with different pipelines may also increase the chance of detecting false positive results, which would reduce the interest of combining sample sizes that we said previously. So what we study, in fact, is the validity of these analyses when we combine data. what we did was performing between group analysis. We want to observe the difference between a first group and a second group, where the first group data is processed with the first pipeline, and the second group data is processed with the second pipeline. And we did this for various pair of pipeline I and J. What we did to test the validity of analysis was creating our groups so that there was no difference between groups. This means that for hypothesis testing at second level analysis, after second level analysis, we put ourselves under the conditions of null hypothesis. And what this means is that if there is no pipeline effect, we expect our results are in fact false positives. And this means that our detection rate is a false positive rate. And what this means is that if we define a specific error rate, for example, 5%, if we have no effect of pipelines, we are supposed to have 5% of false positives. And if we have an increased number of false positives, if we have more than 5% of false positives, this means that combining the pipelines increases the chance of finding false positive results, and so that the combination of pipeline is invalid. So as we said, so to do so, we first define 12 subject-level processing pipelines, which vary on the parameters that we mentioned previously. First one is smoothing. For smoothing, we had two possible parameter options, which were smoothing kernel fluid width at maximum equal either to 5 millimeters or to 8 millimeters. The second one was the presence or absence of the temporal derivatives of the HRF in the design matrix at first level analysis. And the last one was the number of motion regressors. Either we had no motion regressors or we had 6 or 24 motion regressors. So this led us to have 12 subject-level processing pipelines that we applied on data, on subject data from the Human Connectome project, 1,200 subject data sets. And we selected the subject data for subjects who had completed the motor test, for participants who had completed the motor test. And there were 1,080 participants who had completed the motor test. And at the end of first level analysis, we selected a contrast corresponding to activation for right hand. And we did this for all our 1,080 subjects for all of our 12 pipelines. Then, once we had this processed subject data, we performed between group analysis, as we said, between a first group with data combined with the first pipeline, and a second group with data processed with the second pipeline, and we did this for various pairs of pipeline. We observed the difference between group 1 and group 2 and performed a one-tail t-test. We used a family-wise error rate with a thresholding with a correction p inferior to 5%. This way, this means that we expect that there is a 5% chance if we do one between group analysis, that this between group analysis will have at least one significant difference. With this, we define a framework to estimate false positive rates, which is for the pairs of pipeline for which we wanted to test validity. We perform this between group analysis 1,000 times, and we observe the proportion of analyses which had at least one detection over our 1,000 repetitions. For example, if we had, for a given pair of pipeline, 200 out of our 1,000 repetitions which had at least one detection, our false positive rate was equal to 20%. And so this is how we estimated our false positive rates for various pipeline combinations. So I'm going to present the types of pipeline combinations that I will present here I selected for this presentation. The first one were same pipeline analysis. So this is a case where we have no combination of different pipelines. We perform between group analysis where both groups are processed with the same pipeline, and we did this for all of our 12 pipelines that we defined. The second case was the case of different pipeline analysis, which is the one which mainly interests us, and to which we can compare the results obtained for same pipeline analysis, notably, where we used a same default pipeline for group 1 for all our analysis, and for group 2, we used one of the 12 pipelines which was available. And we performed between group analysis between the default pipeline and each of our 12 available pipelines, and observe the false positives for each of these between group analyses with different pipelines. The default pipeline parameters were 5 mm for smoothing, 24 motion regressors, and no temporal derivatives of the href. For same pipeline analysis, here are the results that we obtained. So as we said, we did between group analysis where both groups are processed with the same pipeline, and we did this for all of the 12 pipelines available. So these are the results obtained for each set of parameters corresponding to a pipeline. For example, in the upper left case where we have 1.7%, this corresponds to the false positive obtained where both pipelines had no motion regressors, no derivatives, and 5 mm for smoothing. As we can see here, we had all our false positives which were under the 5% threshold, which means that we do not have an increased chance for any of these situations to obtain false positive results. Then, for different pipeline analysis, here, as we said, we compared the first group with a default pipeline to a second group which was another pipeline, and we tested this for all 12 available pipelines for the second group. So here, for one case, the parameters correspond to the parameters of the second group pipeline. Notably, since we compared the default pipeline to all available pipelines, there was one case for which the second group pipeline had the parameters of the default pipeline, which were 5 mm, no derivatives, 24 motion regressors. And in this case, we had default versus default, which was the only case of same pipeline analysis in this table that we put in gray. We also, I also put in bold the false positive rates, which are over the 5% error rate, and for which we have an increased chance of finding false positive results, which means that we have invalidity. So now we specifically interest ourselves to the results obtained in the case where we have one difference between both pipelines, one parameter which differ between both pipelines, and one case where we have two parameters which differ, which are smoothing and regressors. The first case which interested us was difference in derivatives. So here in green, we have put the case for which the second group pipelines had derivatives in the design matrix, and the other parameter values were the same as the default pipelines. When we have only a difference in presence or absence of temporal derivatives of the HRF, we did not have an increased false positive rate compared to the same pipeline analysis, which was we had a false positive rate which was similar to those obtained previously. So this means that the combination of pipeline in this case with the paradigm that we use seems to be usable in practice. We seem to be able to combine this pipeline in practice without having an increased chance of finding false positive results. Then we observe the results obtained with a difference in smoothing. So here, the second level pipeline only differs from the pipeline on smoothing, where it is equal, where the full width at half maximum of the boosting kernel is equal to 8 mm in the second pipeline. So here we see in this case that we have 5.1%, so which is slightly above the 5% error rate. We can say also that for other comparisons which have only this difference in pipeline that we found in our work, we consistently had false positive rates over 5%, meaning that there is invalidity in this case, and that we cannot combine data due to the effect of pipeline differences. The last difference that we observed was the difference for motion regressors. And we observed that in the case where we had 24 motion regressors in the first pipeline and none in the second pipeline, we had 8.7% as a false positive rate. And similarly, this means that the pipeline combination was invalid. Finally, we observed the results obtained when we combined differences in smoothing and differences in motion regressors. The first main thing that we can observe is that we have, with smoothing and motion regressor differences, we had our highest false positive rates, also when we had the presence of negatives, but we had around the same values, which is around 20%. And we see that it seems that combining these differences leads to a combination of effects which give an even higher increase of the false positive rate in this case. Another thing that we observed to have more detailed observation in our results was observing the differences, observing the distribution of the statistical values obtained at second level. So we took a sample for each pair of for each combination of pipeline. We took a sample of second level analysis statistical values at second level over our 1,000 repetitions. And we observed various things, which are PP plots and the overall distributions. So here are the variants of PP plots that we obtained in this case. So what we did was taking, we say we had a sample of statistical values. We took the associated p-values and the associated minus log of p-values. So the higher the statistical values were, the higher the minus log of p-values were. And we compared the ordered the ordered obtained minus log of p-values to the ordered expected minus log of p-values. So here we have the difference between observed and expected minus log of p-values in function of the expected minus log of p-values with the confidence interval under in the case of verifying the null hypothesis at 95%, and the blue horizontal line corresponds to, in the case, our sample of statistical values follows the expected distribution under null hypothesis, which is the Soutenance distribution with 98 degrees of freedom. We observe the results for same pipeline analysis. So as we can see in the tail, because we use logarithmic values, in the tail we had values which were mostly which were essentially around the 95% confidence interval, and they were not higher. So here, when the graph is higher than the 95% confidence interval, this means that we have invalidity. As you can see here, we do not have any of the lengths over the 95% confidence interval for sample analysis. For differences in motion regressor, so we observed in the tail for both directions, for 24 versus 6 and 6 versus 24, and 24 versus 0 and 0 versus 24. And we could see that specifically for 24 motion regressors versus 0 motion regressors, as we had for the false positive rate, the difference between observed and expected minus log of p-values was higher than we expect under the hypothesis. And this means that the statistical values are higher, which is consistent with the increased false positive rate that we found previously for this comparison. We found the same for smoothing differences with 5 mm versus 8 mm. And similarly, we also found that the combination of pipeline differences lead to a combination of effects with an even higher increase of statistical values in the tail. Also, we observe the overall distribution of these second-level statistical values. We can see that when we have no difference, it fits the expected distribution of student distribution with a 98 degrees of freedom, which is plotted here in orange. And we see that, for example, when we have smoothing difference, we have both an effect on the mean with a shift to the right, which makes the statistical values overall higher, and an effect on variance of the set of statistical values, which have different effects in both tails, of course. In one tail, the statistical values will be higher because of higher variance, and in the tail, they will be lower because of the effect of variance. So we can conclude by saying that we exhibited an effect of pipeline differences, specifically for smoothing and motion reversers, which leads to invalidity of the results, which means that we cannot combine this pipeline without having an increased chance of finding false positive results, meaning that this reduces the interest of pipeline combinations. We can see that this effect was observable on the mean and variance of the distribution of statistical values, and that these effects, the consequences of these effects were observable in the tails of the statistical distributions. We can also say, since we only showed a part of our results, that when we did comparisons of pipelines which had the same single or couple of differences, we found consistent results. We found a consistent effect for differences in smoothing and motion reversers, for example, and combination of differences. And also, here we show the results that we obtained when our subject-level processing pipeline were in SPM, but we did the same with subject-level processing pipeline in FSL, and we found similar results also, as we detailed in the manuscript. Now I will go to present my second contribution, which is about the correction of this pipeline effect. As we said, we found an effect notably for smoothing and regressor differences, and we want to know if there is a way to correct this effect so that we may combine data and not have to care about the pipeline effects because it can be corrected in the analysis. One way that we found to do so was adding a covariate at group level analysis for pipeline differences. So we explained that at first level analysis, we use a design matrix which takes the bold signal and feed it to test regressor. At second level analysis, to perform, for example, between group comparisons, we use another design matrix where the series of contrasts obtained at each position of the brain for each subject is fed to a design matrix whose regressors is characteristics for each of these subjects. This can be a binary information about belonging or not to a specific group. This can be a characteristic such as age. And we can add a covariate, for example, where the characteristic is a binary variable indicating the pipeline. So here what we did was having a covariate where we had 1 for subjects processed with one pipeline and 0 processed with the second pipeline. And what we did to estimate the validity of analyzing the effect of this correction was to perform, to use the same framework with analysis with and without corrections to see whether we had an effect in one case and if this effect was adding a covariate. Because we had one problem, which was that in the previous study, our group difference was confounded with our pipeline difference. The problem is that since our group difference is what we want to observe at second level analysis, if this pipeline difference is confounded, we cannot estimate it and remove it from the analysis. So what we did was performing between group analysis with a heterogeneous hybrid group instead of homogeneous groups. we had, instead of having 100% of subjects in each group processed by each pipeline, subjects from group, proportion P1 of subjects from group 1 was processed with the first pipeline, and proportion P2 with the second pipeline, and same with group 2. This way, we could estimate a pipeline effect using a covariate which was not confounded, with, well, the pipeline difference was not confounded with the group difference. And we did this with and without covariate for correction. This allowed us to compare the results without correction to those obtained previously, to see if with different proportions we observe the same effect as with 100%, and we could compare the results without correction to those with correction to see, as we say, if there is an effect that we can erase or reduce. We did this for various proportions. The first one was 50%. At 50%, we had both groups which had the same amount of data processed with pipeline 1 and pipeline 2. So since we did not have any real difference between the groups, we did not have pipeline effects, so we had nothing to correct. However, at 70%, so here the results are for smoothing differences and regressor differences, which was the case where we had the highest false positive rate previously. For 70%, we saw the effect that we saw a slight version of the effect that we observed in the 100% case without the covariate, and the correction seemed to work well with the covariate for the pipeline difference. Similarly, with 80%, although the pipeline effect was not completely corrected, it was partially reduced by adding a covariate. However, at 90%, we had a large effect which was not corrected and even did worse in the tail. So these are the results that we obtained, and we can say that we had a reduction or correction of the impact of pipeline effect of pipeline differences when we had intermediate proportions. When we had 50%, we had no effect to correct, and finally, when we had 90%, the correction was inefficient. This can be due to two things, mainly, that we suggested. The first one was that we had a low amount of data. We did between group analysis where both groups had 50 subjects. This means that having only 10% of these 50 subjects processed with the second pipeline means we have only 5 subjects in each group processed with the secondary pipeline at 90%, which may not be high enough to estimate the effect. And we also have a higher correlation, and we are closer to the exact confounding that we have at 100%, which may make the estimation also more difficult. So now, to give a general conclusion about our work, we have established a framework where we can test the validity of analysis combining data processed differently, using the statistical properties under the null hypothesis to have a way to estimate the validity of pipeline combinations. We found effects in some cases which led to invalidity. So, which means that in these cases, we may not combine data naively without trying to apply a correction without considering the effect of pipeline, because in this case, we will have an increased chance of finding false positives, which will reduce the interest of larger sample sizes, which is a higher probability to find also true positive results. And we found that in some cases, we may apply correction methods which seem to work well at in some cases where we have different proportions in both groups. Now there is also a variety of possibilities for further works that may be studied. The first one was studying other parameter differences or other paradigms. We focused on 3 parameters, but there is a wide variety of pipeline parameters, which may also mean that the effect in practice of analytical variability may be higher. We also focused on one paradigm, but there is a variety of other paradigms, and so the effect of pipeline differences may be different with other paradigms. For example, we use the block paradigm, but the effect of HRF derivatives may not be the same with event-related designs. Also, we may try to use more than 2 pipelines, and also analysis with where we don't put ourselves under the hypothesis and try and find other frameworks to estimate the validity of analysis. And finally, we can also try and see if there are not other correction methods which may work for correction in some cases, notably those where it did not work well when we tried it. So now there are two publications that I have participated in. One was Open Research Taking the Bits and Pieces with OpenARPANET, where I tested a platform for data sharing, and towards efficient technology data reuse, published in ISB, which contains part of the work done in my first contribution. So thank you for your attention. I'd like to thank you all for coming. I'd like to give special thanks to Pierre and Camille, who directed my thesis and helped me over the last few years to progress and into research. I'd like to thank all members of jury who accepted to review my work, and special thanks to my families and friends who also helped me and assisted me, and to everyone in the team for the good time spent at NPEN.
Okay, thank you, Mr. Xavier Roland, for your nice presentation and for respecting the temporal constraints. The first speaker will be Daniel Barguilias, the first reviewer of your work, and who analyzed in deep your work.
Fantastic. Thank you very much for the opportunity to ask some questions. Thank you, Mr. Roland, for a very impressive thesis. Very clear body of work that's taken on an important new question in the field of reproducibility and MRI research. And I just want to say I was especially impressed with the structure, the clear background that was presented, your understanding of the general field and where this fits in, and also the way in which you approached, in a very structured way, the question, ways of addressing it, and the problematic of analytic variability. Also, congratulations on a very challenging moment for everyone in research and outside of research. Being able to complete this work in a timely manner is very impressive, and so I also wanted to congratulate you on that. My key questions in looking at this work are mainly regarding both future directions and its general place within the field. One of the key questions, I guess, and where I'll start, is one of the obvious use cases would be that you have different research groups which are familiar with different analytic tools. And so my question is, one of the obvious use cases being combining pipelines, even with the same parameters, but across different analytic tools. Does that introduce a kind of variability that would be possible to deal with? Is that problematic? Was that something that was tested here, or would it be possible to test?
Can you repeat? I didn't.
The key question is it possible to combine across different analytic tools? So in this case, FSL and SPM,
yes,
or others.
Oh, yeah, you mean the software packages, for example, yes.
What's that in here? Did I miss it? Or,
yes.
Yeah.
So we did consider this in our work when we performed the intra-software analysis in SPM and in FSL. We found that there were some obstacles because of notably differences across software packages in the way certain steps of analysis are done. So that includes the fact that we had obvious differences, for example, in FSL and SPM, such as the fact that in FSL the registration towards the template space is not done at the same time. It is done after first level analysis, whereas in SPM it's done after processing. And we had also other things, such as the fact that the scale of the data is not the same in SPM and FSL, first level contrast. And because of these differences in estimates, we found that it may be difficult to do the combinations across softwares because the scale was not the same, and there is maybe a variability in scales within or across software when combining them also. So we did think about it when in our work.
Out of curiosity, again, because I believe that might be one of the more, let's say, practically desirable examples of analytic variability. What are the considerations in terms of trying to correct for those aspects that you're describing?
One that we thought about was trying to scale the data, and this is where we found that a problem may appear because of the fact that in SPM we have a bit of this estimate of the overall mean value, and because of this small estimate, we have a bit of variability, and we are not sure whether or not it would be better to try with to apply eventually subject by subject scaling, which would be difficult to do in practice because we would not always have a reference in one case to perform the scale, or trying maybe to estimate a global mean signal estimation and perform a scaling on all subject data, which is the same, which would be more desirable, but which would keep the variability in estimates in SPM compared to FSL.
So I was curious, the work that you presented was using the HCP data.
Yes.
And I'm just curious if there were any, if you looked at these same questions and applied these same analyses to other data sets, and if there are specific considerations that might differ depending on the data set that's used.
The thing is that we mainly we did not try to apply it on other data than the data from the Human Connector Project. The thing is that we mainly considered the Human Connector Project data sets because we had access to a very large amount of data which was relevant to which had the relevant type of content that we wanted to use, and which also allowed us to do to perform, as we say, a 1,000 repetition to estimate the false positive rate with a low degree of correlation across the subjects in the different groups. And this was the main interest, which was that we could repeat analysis over a very large sample. And we did not try to redo it on other data sets. This can be something that can be suggested for further work to see if there are any specificities to the HCP data set. I would not specifically think that there are, because what we did was performing analysis where we tried to verify the condition of null hypothesis, where we specifically had no difference between both groups, no constructive difference between both groups. And I would think that we would likely have the same thing if we had another data set, since we would put ourselves also under the condition of null hypothesis. And if there was a pipeline effect, it would likely also be observable in this case.
So one idea just comes to mind, one question, again, curious about your thoughts, would be, for instance, in data sets where motion might be playing a greater impact. How might the correction for motion in pipelines be differentially affected? Things like that.
Yeah. So we consider various things about this. We notably thought about the fact that we added motion regressors in a study that was with a paradigm which is related to motion, to the motor task. Can you please repeat exactly the point that?
Oh, no, it's just a thought of, in terms of thinking about what might be the different, how the results of your, of your dissertation might be affected by data sets that vary differently from the features of the HCP data. And one might be looking at, for instance, group different studies where the groups have quite different motion features that might be impacted differently by the analytic variability in the pipelines.
So maybe I thought maybe you will make me on this if I am wrong, but when you talk notably about the HCP data set, I was thinking that the variability, the aspect of variability that you mentioned was maybe more about subject variability than about paradigm variability, if we could say, because in the HCP data set there are also the subjects have also performed other tasks, and we could also test the HCP data sets with other tasks to see if we have the same effect with motion regressors and things like that. And this is one thing that can be considered. Of course, we can always find, depending on the paradigm, one specific parameter which is related to this paradigm, and for which the impact of analytical variability may vary depending or not if the paradigm is related to it. And as we say, for example, for the HRF, we could expect that the temporal derivatives of the HRF did not have a major effect in the case of block designs, and that this may not be the case if we have event-related designs. For example, if we want to take another example of things which may vary depending on the paradigm.
Other just general questions I was curious about your thoughts on. Regard more of like the practical applications of these to other questions in the field, and of course, resting state fMRI is a big topic and one of the, say, obvious places where pooling data across sites would be advantageous, where certainly there's a lot of pre-processed data out there. Is it possible to bring that together? What are the unique considerations that one can learn from your work here that could be applied in bringing across pre-processed data?
I think it will be maybe a bit difficult for me to answer this question as I do not know much about resting state fMRI, and I'm not sure if I could bring relevant elements about the types of way this work can contribute in the specific case of resting state fMRI. I think that we can have the same frameworks of the question related to the null hypothesis and the detection of activation patterns. But I would not be able exactly to tell you how we could, for example, consider a framework for in this case, where we try to estimate the validity and correct it with the specific tools of resting state fMRI.
I mean, it does seem that you have everything in place to be able to potentially apply that. I may have misunderstood, but it seems like it could be that you can certainly vary all the pre-processing and evaluate the false positive rate.
It's possible to. What we did was taking advantage of the statistical properties of performing analysis where we verify the condition of null hypothesis.
Right.
So I guess we can eventually try and take this idea to the concept of resting state fMRI, where we consider testing combination of data when we are in conditions where we know that we verified the hypothesis.
Excellent. And so the next question also is about further applications, and this might be a very naive question. So, and I mentioned in the report, you've already seen it, but the consideration of like post hoc meta-analyses on statistical maps. Post hoc meta-analyses on statistical maps made available from prior studies. And I'm thinking of a database like NeuroVault, where you have all of these statistical maps from differentially preprocessed data. Is there any impact from this way of thinking that you presented here on how one could combine those kinds of maps, or what considerations there might be? Again, it may be a very naive question, but it was something that came to mind as I was reading your work.
So I guess the main difference between what is in my work and what you consider
is the fact that in my work, the variability appears before group level analysis, whereas in meta-analysis, you consider variability across group level results. In this case, I would guess that one thing that the main thing that we can say, at least in our case where we try to consider the effect, was that we consider the situation with the self-fundamental where we had a difference between groups, which was at least partially correlated to the difference between the kinds, and it was a situation where we found a major effect. For the differences at group level, one thing that we can do is simply naively considering that analytical variability is a part of the variability in results, as can be other sources of variability, such as intersect, interscalar variability, technical variability. What could be, however, what we could also try, if we have information about processing differences at this level, it would be maybe to try and model it within the models of meta-analysis to combine the results. And but it will require to define a framework on how to do so. But
it wasn't really a fair question. It was just a curious. Okay, that was a perfectly good answer. Thank you. Great. Yeah. Thank you so much again.
Thank you, Daniel. It's the turn now to the second reviewer, so myself. First of all, thank you very much for your supervisor for the invitation to participate to this jury. It's always a pleasure to be at Rennes. And it was also a pleasure to read your manuscript. It's very synthetic and informative too. I have a question of vocabulary to be sure that we are in the same line. You say, rightly, in page 35, that reproducibility is repeating an experiment with the same data, the same pipeline, to be sure that there are no errors in the publication. That is reproducibility. And you have also replicability, that is to use new data to be sure that the published results are replicable, okay? And that's the main point here.
yes,
And it's the main point in general in neuroimaging study. And for this point, you propose to share data, right? So you have good definition in your manuscript, but sometimes you have some bias between replicability, reproducibility, even in your presentation.
maybe,
Agree with that?
yes, I agree with the fact that the questions of nomenclature, the thing is that the first reproducibility was first used as a very generic term for all sorts of repetitions of experiments, and we used in the manuscript these 4 words for the different contexts, which are reproducibility, replicability, robustness, and generalizability. And the thing is that I kept here for the sake of synthesis and to be more synthetic and not to give too much detail because so I agree with the fact that it was mainly a question of time for presentation that that prevented me from giving bigger details about the different types of reproductions of experiments that we can encounter in the literature.
Okay, but we agree that we try to limit false positive.
Yes.
Okay, and you use sharing of pre-processing data. It will have been interesting also to mention that it's possible to share raw data and with specific problems that you don't address in your in your thesis, but it's important to say that.
Yes.
Because it's also possible to do this, to harmonize the data at the source and before
Yes.
processing.
This is it was a main question, which is
Yeah.
why don't we try to address, which has the main point, which is that there are also data which are shared, which are unprocessed, and this is notably the case of the Human Cognition Project.
Yes.
And this is because we have unprocessed data that we could process it different ways. And the answer that we gave to that was the fact that we thought that in the future there can be an increase of already processed data, which is shared. Processed data with transport would be relevant for analysis, and there could be an increase in shared data which are processed because there are questions of privacy and also of sustainability, which could enhance these trends. And of course, we could still have data sets which are unprocessed and perform the harmonization, as you said previously. But we consider the case where we can have to use data which is already processed, and we think that in the future this can be an option. This can be a considerable option for the case of data reuse and data combination.
Yeah, I agree with that. It's important to have these two lines and to have pros and cons in the two options, in fact. And the second point is that to limit false positive, here you are in the framework of the p-value.
Yes.
Okay, but it's also possible to have another statistical framework, in particular, to have the size of the effect, confidence interval, and there are several demonstrations of the fact that some false positive with the p-value are obvious if you look at the size effect in two groups, for example, and confidence interval. And it's a pity that you did not mention that.
Yes. The thing I can say about our work is that there are some constraints which were trying to put ourselves in a situation which is what is currently mostly done in research, and this is also the thing that led us to try having family-wise a rate at 5% and observing the values for that, because in today's research it is still very common.
Yes, but it's not good at all.
Yes, I know that it's not good. This is part of my work also, is to say that there are problems with the values and the p-values, but the idea was to consider a case in which the researcher used the methodological framework to observe the effects that they use mostly in practice today.
I understand.
So this is the thing, which is also the reason why we use specifically SPM. We use specifically, I didn't mention it, but we try to use parameter values which are common in the literature, both for the varying parameters and for the constant parameters in our pipelines, so that we have something which makes sense in practice to someone who will say to himself, what if we combine data hypothesis to do a study the way we do studies commonly in fMRI? So this was mainly...
Yes, I understand, but in fact we have to push our community to report p-value, okay, but size effect and so on. And in your manuscript you defend, and I agree with that, the data sharing. But there are some difficulties in data sharing, and they are not very well indicated in your manuscript. It's not so easy to do because we have to add a lot of meta information, the data, how they are processed, and there is a cost associated to that. We need specific architecture for sharing a lot of data, and not only 50 from one group, but 1,000. And also for processing all this data. And what do you think about this remark?
I think I may not have mentioned it specifically when I addressed the issue of when I explained when I focused on the chapter about data sharing and data combination in the manuscript. But I think I mentioned that there were problems at the origin which caused data sharing to appear very late. And so it's still something very recent in the neuroimaging community. I explained that since it was not a common practice in neuroimaging and in various fields, but notably in neuroimaging, where it's very hard to put a lot of very heavy data in online data sets and on data sharing platforms. I explained that since it took a long time before the types of architectures to data sharing appeared, it was not a common practice because of that. However, yes, I may have mentioned that it is still a problem and there are still limitations. The thing is that in spite of this limitation, there is, however, nowadays enough material to think that it may be time to consider the option of data reuse for new studies and cases of data combination.
Yes, I agree with that. It's just to mention that it's not so easy because of several factors, cultural, ethical, and so on, and hardware problem, for example, to share a lot of that. Similarly to my colleague Daniel, I'm a little bit frustrated by the data indeed, because you use a very simple data set, a block paradigm with motor task. And so, for example, if you try to demonstrate the effect of a pipeline, as you said, in your response to Daniel, for the form of the hemodynamic response, there is no way here, because with a block paradigm, if the form is not very correct, you have no needs for derivative in this case. So it will be more interesting to use an event-related paradigm if you want to show that. You know, with the data you used, it was not possible at all. And also, in the manuscript, you give us very poor information about the data. We don't know exactly the spatial resolution of the acquisition. It's very important for the level of the smoothing, for example. You know, it will be very interesting, as already Daniel said, to use this, your framework on different type of data, but with the HCP, for example, with resting state, for the influence on the correlation matrix, for example, because with a motor task, with such a paradigm with motor task, is a very powerful effect. So you have many influence of the pipeline, processing pipeline. In my point of view, what you can answer to that.
I think I can say that we consider the motor task because it was a situation in which that is well mastered in the case of your imaging. So this would be simpler to using the motor task to do further observation about how correct analysis would be in other frameworks. No, we did not have specifically the time to take advantage of these properties of the vector test, so maybe a little repetitive, but I agree with the fact that, as I say in the conclusion, there is a variety of other types of paradigms and data and, which can be used. We had to focus on one specific situation, and we chose this one because of the reason that I told you. But indeed, I agree with the fact that we could try other situations to see, for example, the effect of the HRF, which is not extremely interesting here. The only thing that we say is that when we have a difference in presence or absence of temporal derivative of the HRF, there seems to be no problem for combination. But
the influence of the framework you propose to add, for example, in the design matrix and information about the processing pipeline used, to demonstrate that, maybe another data set will be more informative, you know?
yes.
So, just my point, huh?
It's something that can be considered. Also, maybe the one main problem once we put ourselves, we chose to use motor test, is that there are practical reasons which constrain us a little bit, which is the fact that having more than 1,000 subjects and having to apply 12 pipelines for processing and post analysis, plus performing 1,000 analysis to try and obtain false positives, both in FSL and SPM. We end up with a very large amount of data, and we're doing the same with another with other data. We also require to perform a very large number of analyses and to have precise estimates, and so this is a bit of a practical reason which prevented us from doing so during the course of my PhD studies. But if had it not been for this, I think we would have considered the option. We would have considered the option of trying other stuff.
Okay, and I'm wondering, here you use the information in the design matrix, and do you think it's possible to envisage the fact that using, for example, a generative adversarial network or something like that, to learn the difference between one pipeline and the other, and to apply the difference to be able, after that, to share, to combine all the data? You know what I mean?
Yes, I understand what you mean. I've actually at the beginning, when I was considering correction options, I was thinking I'm not going to the specific detail of choosing generative adversarial networks, but to learn the correction from the data and apply it a posteriori. The thing is that we first considered this correction method because it was a very in practice very easy to do. It's also a very more simple to apply it in practice, as it does not require to estimate it from a lot of previous existing. We can do it directly once we see if just adding a covariate work. In certain case, we can say now in practice we will make this between group analyses. There may be an effect, but we know we can correct it by simply adding a covariate. It's easier to do in practice.
Of course, yeah.
And but effectively, the question of learning the pipeline effect and trying to correct it is also something that that may be interesting, notably because it can allow to address situations in which the correction did not work well. In our case, such as did not work at all, when we are at 90% or 100%, when there is almost full confounding between the group difference and pipeline difference, and this was one of the. This is one of the things for which it may notably be interesting to consider solution.
Mm-hmm. So, do you think it's a good track to continue your work? Try to learn the difference between pipeline when we want to use pre-processing.
Instinctively, it was the one that I thought about, so I would say that so. And then again, it's a question of time and practical possibilities to do so, but it can be considered for studies.
Okay, thank you for your answers. Now, maybe Sorina, it's your turn.
Thank you. First of all, thank you very much for the invitation. It's a great pleasure and honor to be here. And as my colleague said, I really also enjoyed reading your manuscript. It was very informative and easy to read, and the presentation was also very clear. And you already answered many of my questions, so there are many left. But I will try to with something quite practical, actually, because you've done lots of experiments, lots of work, and you talked about the framework that you've been using. Do you think this framework could be used by someone else, like ourselves here? Is it available somewhere, or could we get access to it?
We shared on GitHub for an article the content in code, and so we put it in the GitHub on CNPensin, so you can access to the code that we use to perform the subject level analysis, group analysis, and estimation of false positive rates on the data. And the pipelines that we used are all available and can be reused. Either the same data or the data if you do the necessary modifications. So.
Wonderful. I think that's really excellent because we talk about reproducibility and variability and all that thing, and I think it's very important to be able to share and use the same framework. So now for a question that may be very naive, but I was wondering, have you considered, or do you think it could be possible or interesting to measure the variability using metrics? I mean, something else than the p-values, to say that you have, yes, so not a statistical measure, but quantitative measurements, such as, I don't know, dice coefficients or about something like that?
I'm trying to think about at which level this can be considered. It's also interesting because it's considering these various levels at which variability results appear. We may try to correct them at these levels, so at the end of first level analysis, at the end of second level analysis. Things that could eventually be done to make further. The first thing that we did to have further observations about our data than the simple p-values, simple false positive values obtained, was making observation on the second level statistical values distribution. Maybe it would be relevant to do comparisons at this level between, for example, as we say, analyses which are done with different pipelines and same pipeline analysis. So any metrics that can allow to observe differences across statistical maps at second level with to compare two maps, one for same pipeline, one for different pipeline, for example. Maybe that's something that we could consider. And then see how we could use it in our case to make estimation of validity, because this is the main thing that we that we wanted to address here. I think this is something that for which the reflection should be developed to see how not only what can we try to use, but how we can use it to answer the specific research question that we want to use.
Definitely, yes. I'm thinking of this because I'm also interested in a lower level reproducibility question, like the one you mentioned, for example, on the OASIS, if you use a different operating system. Of course, order of magnitudes are not at all the same, but I think at some point it would be interesting to compare and to see what variability is introduced at which level and how we can quantify it. Thank you. So I think this is a question that you've already addressed more or less with two other questions from my colleagues, but I also asked myself about studies using the non-null hypothesis, and as you mentioned a few times. But do you think, I mean, this would change quite a lot of things. Do you think the methodology could still apply?
We did a very quick test of trying something which is in a very obvious way, not under the hypothesis, which was trying to do an analysis where for one group we selected one contrast, and for the other group we selected another contrast. So of course we had an obvious good difference. The problem was that for the two that we used, which were false positive rates, they were all very close to 1 because of this, because we had almost systematic detection. And so this is what now the interest of the hypothesis was that the false positive rate was an easy tool to use to make comparisons between and estimate validity or not. If we were to use a framework for which we do not put ourselves under the conditions to verify the null hypothesis, this would require to find other methods of comparing the results obtained with and without data combination and the effect of analytical variability on data compatibility. Of course, we may try something which is less as well, because it's not very common practice to do what we try to do to leave the hypothesis of comparing two contrasts which have nothing to. I may try to do a more reasonable, study which corresponds more to the practical case. For example, difference in gender or in age or things like this. This would be a case that could be considered to test something that is not under the null hypothesis, and then find a way to observe the differences. So this can be on detection of threshold maps and second-level statistical values and numbers coefficients, as you say, notably. These are some. So we say that if we consider doing analyses which are not under the null hypothesis, where we do not put ourselves under the null hypothesis, we need to find a new validation framework. I don't think it's something which would be hard, but it's something for which we should require to think about what we use to estimate validity and compare results.
Okay, thank you. And the last one, maybe, now with the experience that you have, if you had one more year to work on this subject, from the different things you mentioned as future work, which one would you pick? Which one do you think would really help you advance on this topic and help researchers in using multiple data and lower the variability?
Maybe the thing that we're missing, we add out two things. So the first thing, as we say, the changing of paradigms to to try and investigate how the effect of analytical variability varies depending on the paradigm, because it's a big question, because as we said, there are parameters which are related to the type of paradigm that we use, and for which the effect of pipeline differences may not be the same. So I think it's something that would be important to investigate. And as I said, also the question of the hypothesis is interesting, and maybe a bit more focus also on correction methods, because I think there are further works. The main point, in fact, is that we have found, as we could expect a bit, but we have details about what is the specific impact of analytical variability in some cases. But for correction, we see that there are still situations for which our method did not work, and for which we could try other things and try using other frameworks. So yes, maybe it's a thing for which I would be more interested in continuing work, the correction of the impact of analytical variability.
Thank you.
Thank you, Sorina. Now, Monsieur Jean-Marc Gézéquel, have you some remarks, questions?
Yes, of course. Yes, of course. First, thank you very much for inviting me in this jury. I really enjoy reading the manuscript and attending the presentation. I think it was clear and to the point. So maybe a word of my background that is a little bit different from yours. I'm actually from the software engineering research field, and specifically in the viability engineering in the context of software product lines, where we deal with large-scale viability at all levels of a software stack, and where we now have something like several thousand of variables, of configuration variables, leading to sometimes in big system to more than 10 to the power of several thousand different possible configurations. So it's a colossal configuration space. Just for the record, the number of atoms in the universe is estimated to 10 to the power of 80. So it's really, we face huge problem with that. So that's my background, which is not at all in fMRI. And by the way, I learned a lot about this thing, and I think that your work has been really, really important for investigating this issue there. So now my question would be even more naive than the one from the other member of the jury, being that far away from your own field. First, I would like to return to what you has been saying about reproducibility with respect to replicability. I think that even mere reproducibility could be a problem. So I'm wondering whether it's the case in your experience with fMRI, in the sense that what we have now is the fact that because we have several processors, we have different operating systems, we have different compilers, we have different libraries, all of them handling basic stuff such as floating numbers. The way they are multiplied or added is not always the same, and the result might be different depending on the processor, the operating system, the compiler, the options, the way they are represented, and so on. So I was wondering whether even the reproducibility could be an issue with the kind of software that you're running, or that due to the structure, it's not really. I mean, if you run it twice on the same computer, is it going to give you the same answer? And if you're running it twice on different computer, is it going to give you the same answer? So do you think there is an issue here, or is not an issue for that kind of software?
For sort of test-to-test reliability, when we have performed the exact same processing, the exact same data, just at different times, you say?
Yeah.
There can be variations in computations. I did not study very specifically this aspect because I focused mainly on the question of methodological choices. So questions which are related to parameters and operations which are used.
Yes, I understand that. I already understand the focus of your thesis, which was the functional part. But my question is, do you think this is also a problem that needs to be addressed, or do you think that it's not relevant for that kind of studies?
I know that it has been addressed in some cases, not very specifically in task-based fMRI, but for questions of measurements of cortical thickness and other things, other subfields of neuroimaging, and for which there were substantial differences. So I guess that for task-based fMRI, this is also an aspect which should, of course, be addressed. I did not address it specifically, but this is important because, notably, since there are fields of neuroimaging for which substantial differences in results were found because of numerical errors and numerical variations, it can happen in the case of task-based fMRI. So,
do you think your framework? Sorry, carry on.
Okay, maybe the last thing I can say about the low-level reproducibility that I tried is that when I tried using software differences, so from one time with FSL and one time with SPM, we found, of course, not the exact same result, but substantially the same results. So I would guess that the variations related to just numerical errors in operating system conditions would be quite lower than the differences we found between FSL and SPM. However, yes, it is something that must be addressed because it's one type of variability which is related, one subfield of analytical variability, which may have an impact on the results.
Yeah, thank you. But do you think that the framework you're proposing could be extended into that direction?
Yes, the main problem, if I take just the question of test such reliability, there is a possibility to try and redo the exact same experiment at a different time, reusing the data and protocol, and try and see if we obtain the exact same results. I guess that there should not be very important differences because it's about a repetition of 1,000 statistical between group analysis. So there will need to be a very large number of differences within a large number of subjects to see and observe large differences at the end, which is the false positive rate. And I don't think with our framework there would be a very large propagation of error and amplification of them, which would lead to very large differences in results. And then there is a question of compatibility between data, and then one thing that can be tried also is to try to do the same as we did with between group analysis with two pipelines, with one pipeline under specific operating system condition and the other one under other specific operating system condition, which is a type of use case of our framework, which would be the most obvious for the specific question that you asked, I think.
Okay. Then another question on your framework, which is basically what you have shown is that you studied basically three variables, and two of them have two values and one has three values, overall yielding 12 possibilities. So I'm wondering if you want to study more variables, more variation points, basically, that have more values. Of course, you have a Cartesian product of all the possible configurations, so it grows very fast. And so I'm wondering how your approach would scale with respect to that. Do you think it could work for, let's say, 100 variables, each having something like between 2 and 20 possible values? Or is there a scalability issue?
I don't think I understand quite well the question. What is respected?
Well, it's when you have shown the results, they were the variables that you were using. Yes, the next one, maybe that. Yes, because after that, when you show the result, the numerical results that you get, maybe you can move on a little bit.
The results after what?
Yes, the numerical results after, yes.
Yes, after that. Yes, again. Yes, yes, this one. Here, that's what I call the variable, and the fact that you have is using smoothing either 5 millimeters or 8 millimeters. So that's one variable with two values. And then derivative or no derivative, that's another variable. And then the third variable is the number of motion regressors, 0, 6, or 24. So you have basically three variables. Two of them have two values and one has three values. So it yields a total of 12 possibilities. So my question is, if you have more variables having more possible values, whether your approach would scale with respect to that? Because I mean, it's normal that for your PhD thesis you concentrated on a few variables to make it clear that how was the problem, what was the methodology, and so on. But my question is, if we would like to apply that in practice for real, we would have many more variables with many more values, and so could we use the same approach?
I guess what you think about is the fact that inevitably, if we have a very large number of variables and varying variables and parameters, and we end up with an exponentially increasing number of combinations, we end up with there is the question of correction for multiple comparisons. I guess it's the progress. And of course, notably in this case, I guess that if we had not only we can also talk about the fact that maybe some of these parameters also have an effect in the case of same pipeline analysis, because this was something that was studied, and this was one question which was addressed notably in various works about analytical variability. Of course, if we had, for example, 1,000 possible combinations of parameters, we would have a very increased chance of finding one which has false positives. For example, in this situation, even if it was a false increase of false positives, if it was a problem due to the comparison, then I guess we would have to observe something else and just a single smoothing, single false positive obtain singularly, and I'm not sure it would. The thing that could be done would be to try and find the confidence interval over our error rate, around our error rate, and correct them for the multiplicity of analysis. There are also other things that we can observe, which is the consistency of effects for a same pipeline difference over our analysis. For example, if we have more than 5% in one case of a difference, for example, a single pipeline comparison, which give 5% false positive rate, we observe all combinations of pipeline which also have this difference to try and find synthetic results and have more precise estimate of the effect of pipeline over this list of over this repetition of analysis with different combination of pipelines, and this can be used to have more precise estimate. Because here we have only 12 plus 12 false positive rate, but we had 144 12 times 12 comparisons of pipelines, and we can typically try and do that with an even higher number.
Okay. And maybe my last question would be to return to one of Serena's question, which is a different way of presenting this, because as an external user of that kind of software, probably the big thing now is that I'm not just interested in getting an answer, because basically the software is running a kind of estimation and giving me some kind of answer. But I would like to see that answer qualified with a confidence interval somehow, or which is the fact that in nature, when we measure something, you never get crisp values. You always get something with a tolerance. And I'm wondering whether you could basically use this information about the variability that you have to bring back an estimation of the confidence of the result you would get from the functional point of view of your application. So I would like you to comment on that, on how it could be used.
To use confidence interval in the case, for example, of false positive rates here.
Yes.
We considered this at one moment, which was that we have what we have here under the hypothesis is we expect to have a 5% chance of having a detection for a single analysis. So this is simply Bernoulli law, and when we do 1,000 repetition, it's a binomial law with p equal to 5% and 1,000 repetition, which can be used. This has been used also in other works which proceeded a bit like ours, like in one work, metal or cluster failure, and they plotted the confidence interval over the 5% error rate. So here what we found was that we always found false positive rates which were below the 5% confidence interval in this case, which we could expect because in the literature there was information about the fact that using smoothing, notably with SPM, with the random field theory at second level, using smoothing plus random field theory could lead with small levels of smoothing, or levels of smoothing which are around ours, would lead to lower false positive rates, to lower probability of detection. So, which is the case here, since we have a values under 5%. The main thing that we wanted to do, the main reason why we did not interest ourselves specifically to the 5% confidence interval, to the 95% confidence interval over our 5% error rate, is that we did not consider that it was really problematic if the false positive rate estimated were below 5%, because it would not result in an increased chance of finding false positive results, which is the main problem in the case of data combination. However, when we had a large increase over the 5% error rate, we considered that it was a case where we could not combine the data in practice because it would be problematic in the situation that we want to address.
Okay, so thank you very much, and again, congratulations for your work.
Thank you, Jean-Marc, for your intervention. Now we have the two supervisors of your work. Camille, do you want to add some words?
I'll say a few words. If you don't mind, I'll switch to French.
Yes, if you, of course.
J'écris quelques mots. Xavier, après 3 ans et demi de Champagne, je suis vraiment heureuse aujourd'hui de devoir soutenir ta thèse. Quand nous t'avons rencontré avec Christian, c'était il y a un peu moins de 4 ans. Tu terminais un stage en mathématiques de la musique et avec nous, tu as accepté de te lancer un nouveau défi à la découverte du cerveau avec la neuroimagerie. Ta thèse n'aura pas été un chemin sans obstacles et je pense que le reste de l'équipe se joint à moi ainsi que le jury en pensant à Christian aujourd'hui qui nous a quittés au milieu du parcours de ta thèse et qui nous manque alors que tu t'apprêtes à franchir la ligne finale. Et puis comme pour toutes les doctorants et toutes les doctorantes ces dernières années, tu as dû apprendre à mener ta thèse en composant avec le travail à distance, les incertitudes liées au Covid. Mais avant tout cela, cette thèse t'aura quand même permis de découvrir un bel aspect de notre travail avec les missions et de voyager. Je me rappelle notamment de ta première conférence, un hackathon à Paris, où on était partis ensemble. Et on est bien étonné de découvrir en arrivant que pour entrer dans la salle principale de la conférence, il fallait enlever ses chaussures. Donc, ça t'aura permis de découvrir tes premiers neuroimageurs en chaussettes, mais aussi d'échanger sur ton travail, qui, je crois, est un aspect que tu avais apprécié. Tu as ensuite eu la chance de découvrir le soleil marseillais autour de la science ouverte et même un séjour à Rome. Donc voilà, tu es presque arrivé. Bon, c'est pas encore fini, mais je terminerai quand même en te félicitant d'être arrivé jusque-là. And I don't have any questions.
Merci.
Merci. And now, Pierre,
Okay.
supervisor and director of the team now, where you have worked during 3.5 years.
Ok, moi je vais faire en français aussi. Merci Xavier pour la présentation, pour les années passées ensemble. Merci aussi aux collègues du jury d'avoir accepté d'être là. On se disait ce midi que ça faisait très longtemps qu'on n'avait pas fait une soutenance en présentiel. C'est quand même très agréable de retrouver ça. Donc, ce qu'on m'a dit Camille, une thèse, c'est jamais un long fleuve tranquille. Mais toi, c'est vrai que tu as un peu cumulé les contextes un peu difficiles avec le décès de Christian, le Covid. Et c'est vrai que malgré tout ça, malgré une longue période de télétravail, là, je me rappellerai longtemps de nos réunions en visio où on parlait pendant 30 secondes et puis on devait s'arrêter 30 secondes parce que ton voisin avait décidé de mener des lourds travaux pendant le confinement. Donc, il fallait attendre que la personne s'arrête avant de pouvoir continuer la réunion. Voilà, bon, c'était folklorique. Mais bon, malgré tout ça, tu as quand même réussi à mener un gros travail d'expérimental dans un cadre original avec de software là, qui ont permis de faire avancer la compréhension de cette variabilité induite par les pipelines. D'avancer dans la direction d'utiliser ces données hétérogènes. Donc, félicitations Xavier. Déjà d'être arrivé là, malgré toutes ces difficultés. Et puis bon, pour finir, je sais que dans la suite, là, tu vas te diriger probablement plus vers l'enseignement, que tu as un peu fait aussi pendant ta thèse. Donc, bien entendu, je te souhaite le meilleur pour cette nouvelle aventure. Peut-être pour finir pour une note un peu plus légère, bon, le problème c'est que tu vas manquer à ton équipe du quiz, du Saint-Y, de lundi soir, qui perd un élément majeur. Mais bon, de l'autre côté, ça laissera un peu plus de chance aux autres. Et bon, comme on dit en Bretagne, bon vent, et puis peut-être à bientôt quand même, le lundi soir. Voilà, merci à toi Xavier.
Merci Pierre. Alors, traditionnellement, y a-t-il des questions dans l'audience de quelqu'un qui a au moins le titre de docteur? Pas de questions. Bon, merci pour cette présentation, merci pour votre écoute. Et
on va rester là, ouais.
Il va falloir vous retirer pour qu'on puisse délibérer en place. Merci. On vous dira, ça va pas durer 2 heures.